All right, guys, so this week we're talking mutations, mutation repair. We're kind of taking last week's genetics talk and we're going to expand on that a little bit. The first thing I wanted to talk about, though, was gene transfer. So vertical gene transfer is normal reproduction. So when you have generate new offspring via either binary fission, if you're a bacteria, or mitosis, if you're in a eukaryotic cell, that's vertical transfer, going from a parent down to a daughter. Now, if you transfer genetic material between two cells, well, that's called horizontal gene transfer. And usually we're talking about bacteria in this case. And so conjugation would be the example of like a sex pilus or a pili. Multiple ones would be called pili. And that's what we've talked about before, where we've talked about sex pili, where conjugation is when two bacteria connect to each other via a pilus, and then they can transfer usually a small piece of genetic information in the form of a plasmid, which is a really small circular piece of DNA that only has maybe a couple genes on it. But that's how they could transfer antibiotic resistance between two living bacteria. Now, something that we take advantage of when we're trying to do biotechnology would be transformation. So that's when we have a bacteria that will take up DNA, I say here from a dead buddy, but it could be just any random DNA that's sitting on the ground. And so what we do when we're trying to do biotechnology is we can just artificially give them a piece of DNA. And so you can generate a plasmid, and you could put that in the same culture as like an E. coli, let's say. And that E. coli will take up that DNA and then incorporate it into its own genome, or at least as a plasmid next to its genome. It can also do that if a nearby bacteria dies and leaves its DNA around. So transduction is the third way. It's not as commonly talked about, but transduction would be when a bacteriophage, which is a type of virus, we haven't talked about viruses yet, we will, and we're going to talk about phages and phage therapy. But basically it's a virus that takes over a bacterial cell, and when it does that, it makes baby viruses inside of the cell, and then they burst out and explode. So if that happens, sometimes those baby viruses, as they're building their own DNA to go in the new baby viruses, sometimes they can accidentally incorporate some of the host cell's DNA, and then that can get transferred to new cells. And so that's called transduction. We also do that in the laboratory. If you need to transduce something using a virus, like an adenoviral vector, if you're trying to do gene therapy. So that term basically just means that you're using a virus to move DNA, as opposed to artificially just forcing the DNA into a cell using transformation. Now that we have that out of the way, I want to start talking about mutations and mutation repair. So let's talk about the central dogma again. So the central dogma is when we take our DNA and we convert it to messenger RNA, and we convert that to our polypeptide chain via translation. Okay, so DNA to RNA is transcription, RNA to protein is translation. This should look familiar. So we start in the center using either our RNA transcript, which is just our string of nucleotides that make up our RNA, or our coding strand, which those should look almost identical, except instead of thymine in our DNA, we have uracil in our RNA. So you start in the middle and work your way out. Now, a mutation is just when we change what those nucleotides are. And that can either not be a big deal, we call those silent mutations, or it could be a really big deal because it's going to change the amino acid. So if we start in the center here and we say, let's go GAC, that would make aspartic acid. Okay. Well, if I mutate that G from my codon, remember codons are 3 nucleotides, so if I mutate that G to a uracil, so now it goes UAC, and that would make tyrosine. So that's a different amino acid. And we know that if you change the amino acids, you're changing the primary structure of a protein. That could have a huge effect on the functionality of the protein. It might not, but it really could. And that's what's really dangerous about mutations. So mutation is the change in the order of nucleic acids. Usually we think about them in terms of being harmful. They could be neutral. So like if you change your amino acid and maybe it doesn't really make a big difference, or maybe you change the nucleotide and you end up not even changing the amino acid. That would still be a mutation, but they could be neutral if they're not really doing anything. There are hypothetical beneficial mutations. The best example of that would be antibiotic resistance in like a bacteria, where they're able to mutate maybe where an antibiotic would normally bind to, let's say, penicillin binding protein, and you can mutate that penicillin binding protein so that penicillin won't bind to it and won't break it. So that would be a beneficial mutation. Viruses also engage in lots of beneficial mutations. Beneficial we usually consider as being something that helps the organism fend off some kind of selective pressure. Now, the causes of mutations can vary, and we're going to talk about those today. Usually we think about it in terms of radiation. That's what most people think about as causing mutations, but there can be chemical versions. There's actually some examples inside the human body that we purposely engender mutations in certain genes to increase diversity in those genes. But actually, the most common source of mutations is from DNA polymerases, which normally they're just going to make the new strand of DNA during replication, if that DNA polymerase makes a mistake. And so you can see here, we call that a mismatch, and usually our DNA polymerases will fix it. So in this case, the DNA polymerase, which is that big green box, is moving along. It's building from the 5' to 3' direction, right? It adds new nucleotides onto that 3' carbon. And in this case, it was supposed to put a thymine, because this is making DNA, right? And A's match up with T's. And it was supposed to put a thymine, but it accidentally put a cytosine. That's not good. And so what happens is, in the case of most eukaryotic polymerases, and some bacterial polymerases as well, they do this kind of fact-checking. That's what I like to call it. And it will go back and look at any mistakes that it made and make sure that the nucleotides are lined up correctly. And in this case, it sees, oh, I accidentally put a cytosine instead of a thymine, and so I'm going to cut out that cytosine and put a new thymine in. We call that exonuclease activity, and we're going to talk a little bit more about that. Now, usually what we're talking about are point mutations. A point mutation is just when we take a single nucleotide, so your A's, your T's, your C's, your G's, and we change those to a different nucleotide. And so this picture on the right here, you can see we're going to start out our DNA strand is a TTC. And so in this case, we're actually going to be mutating the template strand, but it doesn't really matter. We're just, that way our messenger RNA lines up with our DNA. So in this case, we'd have thymine, thymine, cytosine, okay? And so the matching RNA would be AAG, or adenine, adenine, guanine. And that makes the nucleotide called lysine, which is an interesting amino acid. If you take a biochemistry course, we actually go into more depth about each amino acid. There's a bunch of different kinds of amino acids. And for this class, you don't need to know all of your amino acids because I will be giving you the chart that you need, i.e. this chart, but you just should know that the R group of the amino acid is what changes. And so your amino acids will always have this same structure on the bottom here, but we actually end up changing this structure up here called, it's our R group, but basically any kind of chemical structure, if we change something or can change something, we call that an R group. So if we have a point mutation, so we're mutating one nucleotide, let's say, for instance, we change that cytosine to a thymine. Okay, well, let's go back to our original chart here. And so if you look over here, we're looking at our RNA, right? And so it was A, G, that would code for a lysine. That's our amino acid. And if we mutate that third nucleotide to go from A to A, it still makes a lysine, and so that's that example of a silent mutation. Now, we've talked previously, certain amino acids that we make actually don't code for amino acids. They code for stop codons because you need to tell your RNA polymerase when to stop making the messenger RNA. And so, for instance, let's say instead of AAG, we're going to mutate that first nucleotide in our codon to a uracil. So let's take a look at our chart here. So we were making AAG, but now I want to make UAG. That codes for a stop codon. So a stop codon would be what we call a nonsense mutation. If you accidentally make a stop codon too early, what that's going to do is that's going to make your protein too short. We call that truncation. And so if you truncate a protein, you've probably drastically changed its functionality, if not completely annihilated it. Because what if you had a stop codon occur after only maybe 20 amino acids? That's probably not long enough to actually do anything. And so you're left with this truncated, useless protein. And so that's called a nonsense mutation. So now let's look at missense mutation. So if it was AAG, let's do AGG. Okay, so let's look here. AG makes lysine, right? So now let's do AGG. It makes arginine. So let's take a look at arginine. So here's arginine right here. And arginine also has a charge. So something that we haven't talked about yet, and something that I'm not going to emphasize too much, but just so you understand this idea, is that amino acids can have charges. They may not have charges. It just kind of depends on the amino acid. If you have a positively charged amino acid and a negatively charged amino acid, they could stick together. And so that's how you can get secondary and tertiary structures of proteins, which we've talked about before. So in this case, I have a missense mutation. So I've mutated this nucleotide from an adenine to a guanine, but what it's resulted in is a different amino acid. That is true. But the trick about lysine and arginine is that they both have a positive charge. So you can see here there's a positive charge right here, and right here there's still a positive charge. We call that a conservative missense mutation. Missense just means that I'm still making an amino acid. Most of the mutations that we're going to be talking about, they're either silent, so you don't even notice them, or they're missense. So you're still making an amino acid, and your protein would still be the same length. You've just changed one of those amino acids. And that's a way that we can study proteins. So we can purposely change amino acids at one specific site on that protein and see if that changes the functionality of the protein. But if we're maintaining that amino acid's general vibe, if you will, so is it positively charged? Is it negatively charged? Is it nonpolar? Is it slightly polar? Things like that. Then that means it's conservative. It's conserving that trait of that amino acid. And the opposite is also true. For instance, if I accidentally make a threonine, I won't go back to the chart, but if I make a threonine here, that has a very different structure. It's a non-polar amino acid. And so in this case, we've gone from lysine, which is polar, or it's charged, as a positive charge, to threonine, which is non-polar, or at least slightly polar, and that's really, really different. And so what that's going to do, it's not the same kind of amino acid. It doesn't have a nice big positive charge on it. And so what does that do? Well, we call that a non-conservative mutation because it's going to change that amino acid's abilities. So for instance, if my secondary structure relied on this lysine sticking to a negatively charged amino acid, well, and then I've mutated my lysine to a threonine, which doesn't have that negative charge, now that secondary structure might fall apart because I don't have that nice little magnetic attraction between a positive and a negative charge. There's another set of terms that get tossed around. I don't really like using them, but it's the terms synonymous and non-synonymous. Just in case you encounter these terms in the future, that's why I wanted to bring them up. So a synonymous mutation just is a silent mutation. That's basically all it means. It means that you've resulted in nothing that really matters. Non-synonymous mutations usually are changing your protein. And so the mutations themselves, like a point mutation is referred to as silent, nonsense, missense, whatever. Non-synonymous refers to more the protein itself. So is the protein sort of broken or is it not broken, i.e. synonymous? Now, when we are looking at these nucleotides, there's two different types of mutations that we can actually see for these point mutations. So am I converting between two different purines? Okay, because we talked last time that there are purines, those have 2 rings, and there are pyrimidines, which only have 1 ring. So your cytosines and your thymines and your uracils are pyrimidines, and your adenines and your guanines have 2 rings, and so those are purines. And so if I'm going between an adenine and a guanine, they're both purines, and so we call that a transition. It's transitioning between these 2-ring structures, i.e. they're not that different. Yeah, there's some different groups on there, like there's this double bonded oxygen or this nitrogen here, but for the most part, they still have 2 rings. Now, if you converted like an adenine and basically got rid of this entire ring, then we call that a transversion. It's a bigger deal to go from a purine to a pyrimidine and vice versa. There's a bunch more atoms that you either need to add or take away. So these are called substitutions as well as point mutations. It's the same thing. But you can also have a different kind of mutation that's called a frameshift mutation. A frameshift mutation is when you either add or delete 1 or 2 nucleotides. And if you do that, it's going to shift the entire codon reading frame. The codon reading frame is just what we call those sets of 3 nucleotides that form codons. And they're supposed to be very, very consistent so that your ribosome can look at those 3 nucleotides and then make that amino acid or start building the amino acid chain properly. So if you shift that whole thing over by adding 1 or 2 nucleotides, then that's going to change completely the subsequent set of codons. And you could accidentally make a Nonsense mutation, that would probably be the worst. So you could truncate the protein, or you're just going to completely change all of the subsequent amino acids. And so it could be really, really different. Maybe you have a lysine and a proline and a glycine, and now they're all just completely different amino acids, and it's going to completely change your protein. It falls apart, it doesn't do its job, and that's really, really bad. Something tricky is that frameshift mutations with 3 nucleotides that are added or 3 nucleotides that are deleted is not called a frameshift because you haven't actually changed the subsequent amino acids based on their codons, because those are all still there. You've either just completely deleted one amino acid because there's 3 nucleotides per codon, or you've completely added one new amino acid because you've added 3 nucleotides, which is another new codon. And I know that's kind of confusing, like it does seem like you're shifting the frame, but the actual amino acids are what we care about. And those amino acids haven't changed if there's 3 nucleotides added. But for my money, I would say adding 3 of them is still an addition or a deletion. It's just not a frame shift. Now, mutations exist in basically every kind of creature ever, including humans. And if we look at mutations on a population level, then we can get some really interesting information. And so lots of humans have lots of mutations. You're full of mutations. If it's a single nucleotide that's changed, we call that a single nucleotide polymorphism. And what we call that is a SNP. So if you hear that term talked about, that's called a SNP, single nucleotide polymorphism. And what that really means is that at least 1% of the population that you're looking at has that mutation. And so you can look at people's whole genomes and you say, okay, well, 99% of the population or 95% of the general population has a G at this specific site in its DNA, a G and a C. Well, if maybe a 5% or 1% of the population has a thymine and an adenine at that site, it could be higher, then we call that a single nucleotide polymorphism. And so it's really more looking at the population level, but you can see an individual that has that SNP as well. And that also applies to the actual DNA as well. And that's what this bottom right diagram is talking about, where there's synonymous and non-synonymous again. So synonymous means basically that nothing is really happening to those proteins. But a non-synonymous mutation, i.e. a non-synonymous single nucleotide polymorphism, basically means that you're messing up those proteins. Either you're truncating them and creating a nonsense protein, or you're changing those amino acids, which makes a missense protein, which could also mess things up. There are plenty of examples of non-synonymous mutations in populations. They don't necessarily have to be fatal or even that detrimental. And so the idea of a antibiotic-resistant bacteria that can still be a non-synonymous missense mutation, right? You've changed that protein so that penicillin maybe can't bind to it anymore, but that doesn't mean that it's actually hurt. It just means that it's got some kind of non-synonymous missense mutation and it's still functioning properly. Now we need to talk about where mutations actually come from. It's a really good question. So most of the mutations, as I mentioned before, are caused by DNA polymerase making an error when it undergoes replication. And so when they make those kinds of mistakes, that's not a good thing. And so most of the DNA polymerases, at least in the higher order organisms, i.e. even bacteria, you could argue, but pretty much all eukaryotes do this. Most bacterial DNA polymerases also have fact-checking ability. And when I say fact-checking ability, that's really more often called proofreading, and that's the exonuclease activity that DNA polymerases engage in. That's also called 3' to 5' exonuclease activity, because normally DNA polymerases build from 5' to 3', right? And so they kind of look back from the 3' to 5' direction, the other way. And that's where that name comes from.
Proofreading polymerases have several checkpoints to prevent incorrect nucleotide incorporation during the DNA extension process. The first checkpoint is a result of the enzyme's significant binding preference for the correct versus incorrect nucleoside triphosphate during polymerization. If an incorrect base does bind to the active site, incorporation is slowed, increasing the opportunity for the incorrect nucleotide to dissociate and a correct nucleotide to bind. Alternatively, if an incorrect nucleotide is incorporated, the proofreading polymerase detects the perturbation caused by the mispaired bases and shifts the 3' end of the growing strand to the polymerase's proofreading exonuclease active site domain, where the 3' to 5' exonuclease activity removes the mispaired base. With the mispaired base removed, the polymerase shifts the strand back into the polymerase domain and continues adding bases and extending the DNA.
how well a polymerase, usually we're talking about DNA polymerases, how well it can actually fix any problems that it causes, that's called fidelity. And so if we're doing research and we need to do PCR, which is just where you make a lot of DNA, you don't want mutations to happen. And so we call those high fidelity DNA polymerases because they're really, really good at making sure that they don't make errors. Now, there are examples of RNA polymerases and DNA polymerases that make a lot of mistakes. We tend to see those in creatures that want to have mistakes occur because that allows them to evolve very rapidly. The best example would be in viruses. Lots of viruses make a lot of mistakes when they're actually replicating their genomes. But it's to their benefit because it's all about numbers. The more often they can make mistakes, the odds are that they will happen to make viable offspring that has a mutation in something that could allow it to avoid some kind of selective pressure. So a great example would be HIV. So HIV has a DNA polymerase, i.e. its reverse transcriptase is the enzyme that HIV uses, but it has DNA polymerase activity, and it's really, really bad. It makes mistakes all the time, but HIV loves that because it will accidentally make offspring that have genes that are mutated inside the HIV that allow those proteins that those genes make to be resistant to drugs. So if you put a patient on a protease inhibitor, then you might mutate your protease so that those protease inhibitors don't affect it anymore. And that's a byproduct of this rapid or high amount of mutation that can occur because of your DNA polymerase activity, or i.e. not being able to fix the mistakes from that DNA polymerase activity. You have a lot of mistakes from your own DNA polymerase, but it fixes most of them. But we also know that there are other ways that you can have mutations occur, right? So radiation is one of the most common ones that we think about. So that would be like UV light on the right side, which can cause thymine dimers. And that's just where two thymines are basically stuck together. And so when your DNA polymerase or your RNA polymerase are cruising along that strand, they could accidentally skip over that nucleotide, or they could accidentally match it up with only one adenine instead of two, and that would be really bad. We have ways of fixing that, which I'm going to talk about that in a minute. Now, ionizing radiation, or also free radicals can do this. They basically cause so much energy to be put into the atoms that comprise your DNA, especially in the backbone, that they excite the electrons of the atoms in those backbones, and they excite them so much that they start reacting just with random stuff nearby. And so what that can do is it can cause the covalent bonds that are supposed to be forming this nice backbone, it can cause those covalent bonds to break and react with other things. We call those, if it happens on one side, a single-stranded break. Or if it happens on both strands, we call that a double-strand break. And those are absolutely the worst ones that can happen. And the more powerful or the more energetic the radiation or the more powerful the free radicals that get exposed, the more possibility that there can be a double-stranded break. And so that's why if someone gets really severe radiation poisoning, that's what's happening. Now, you do have ways of fixing those problems. We have ways of fixing all of these problems. But sometimes if there's just too many double-stranded breaks, we don't have the enzymes available, or at least the number of enzymes available, to fix all of those problems, and so the cells die. There are also examples of chemicals that can cause these kinds of mutations. Oftentimes what the chemicals will do, they will either kind of stick in your DNA and look like another nucleotide, and so your DNA polymerase or your RNA polymerase might think there's an extra nucleotide there, so it would act like an insertion. Or it can change the nucleotides that are there and make them look like different nucleotides, i.e. like a substitution. There are a bunch of examples of this. I have a couple charts here that list a few of these, but they're just chemicals. We have other uses for them, but you just don't want to spill them on your skin. Now, I mentioned earlier that there are some examples of intentional mutations being created inside your cells, or any kind of cell. Usually that involves antibodies, and so if you've seen an antibody before, this should look familiar. If you haven't seen an antibody before, basically this is the business end of antibodies. We've all heard of antibodies, right? They're what your B cells secrete to grab onto bad guys, and this is where a bad guy would get grabbed. Okay, and so this area, i.e. the variable region, that's what's in red, the variable region of your heavy and light chains, because there's a heavy chain and a light chain to just kind of get you ready for immunology when we talk about that. So this variable region here, we want that to grab onto essentially any kind of antigen or any epitope on any antigen in the known universe. And so we need to be able to mutate this section of protein right here to potentially bind to anything, literally anything. And so we engage in something called somatic hypermutation. And there are special enzymes, usually cytidine deaminases, that are able to change the actual DNA sequence that codes for this protein, thus changing the actual structure of this protein. I don't want to spend too much time on this, but I mentioned before there can be beneficial mutations, there can be harmful mutations, there can be neutral mutations. We can define our mutations not just on what's physically happening, but on what kind of effect they have on the creature itself. And so you can gain function, you can lose function, it could completely kill the creature. It could just be kind of harmful to the creature, i.e. it makes it, usually what we're looking at is reproductive ability. So is it making more offspring or allowing those offspring to survive? Or is it not making more offspring or making it less able to care for its offspring? So beneficial would be a good thing, harmful would be a bad thing, of course. Neutral just doesn't really make a difference. Nearly neutral is kind of a tricky one, but basically if you look at the creature and the ones with this mutation are a little bit better, it's not necessarily super beneficial, but it's maybe a little bit better, then we call those nearly neutral. But you can see here on the right side, there's lots of different ways to actually classify mutations. So an example I like to talk about is with viruses, because I'm a virologist by training and an immunologist by training, but viruses can engage in mutations. As I was mentioning before, a lot of them have really, really bad functioning DNA polymerases, but that's actually a good thing for a virus because it wants to have lots of mutations. And so you can see here in the red, those are RNA viruses, in the blue are DNA viruses, and down here is the mutation rate. So the bigger the number, the more often you have mutations. And you can see here lots of viruses, including HIV, which starts with an RNA genome, they have lots of mutations that occur, which means they're able to mutate their offspring much more easily, and that makes those offspring potentially able to resist some kind of selective pressure, i.e. some kind of drug. And for example as well, you can see over here, we've got every kind of creature depicted here where you've got the size of the genome, so we would be way over here, and then you have your lower division eukaryotes, you got your bacteria, your archaea, and then you've got lots of different kinds of viruses. And so you can see there's also a correlation between the size of your genome and your propensity for mutations. And so you can have lots more mutations occur in like your RNA viruses, which is what's matching up here, versus your DNA viruses, because the bigger your genome gets, the harder it is to actually fix those problems. And so you don't want to, cause too many mutations because you have a huge genome. So if you have too many mutations, that's not a good thing. And so that's why, based on genome size, you have a reduced mutation rate, i.e. you have more mechanisms to repair those mutations. Now, speaking of mutation mechanisms and the mechanisms of mutation repair, let's talk about that next. And so you can see on the top here, this would be all the different kinds of ways that you could accidentally cause some kind of mutation, be it a frameshift, be it a point mutation. It doesn't really matter. There's all kinds of different ways. Maybe you're talking about a strand break. And these are all the different ways. And then you can see underneath each of those ways are the mechanisms that exist inside of cells. Usually we're looking at eukaryotic cells, but these are mechanisms that exist to fix those problems. And so I want to go through each of these and address how we actually deal with these things. Now, the first one is one that we've already talked about, and that's replication repair. That's that 3' to 5' checking that a DNA polymerase does. And so when it accidentally makes a mistake, it can then go back using its exonuclease activity and fix that nucleotide. And so in this case, it's accidentally put a thymine here, which thymine does not match up with guanine. It's supposed to be a cytosine here. And so it recognizes that problem. And so it's been building from 5' to 3', but it's fact-checking itself or proofreading itself from 3 to 5. And it says, hey, thymine doesn't fit there. And it really does that because it detects it because the angle of the atoms isn't correct, because you would have the incorrect number of hydrogen bonds occurring between a guanine and a thymine. And so it gets bent kind of weird. And so it recognizes that that's a problem and it cuts out the thymine, and now it can put the cytosine there. Most of the time, you're actually going to be able to fix most mutations that occur this way using this mechanism, this proofreading mechanism. But let's say, for instance, that you didn't fix it and one of these accidentally sneaks through, which can happen. So then we switch to something called mismatch repair. So mismatch repair is when you have that mutation occur. Okay, so this is really early on following replication, because that's usually what we're thinking about. And so it turns out when you make new strands of DNA, you put lots of methyl groups. Methyl groups are just carbons with 3 hydrogens attached, and you stick a bunch of methyl groups onto the strand or the sugar phosphate backbone of your DNA strands. The ones that exist inside the cell for at least a couple minutes, they've existed for a couple minutes, you start methylating them. Okay, so you have that little window of a couple minutes where the new daughter strand, so that would be this top strand here, isn't methylated. And that tells these special mechanisms or these special enzymes that can detect these problems. They say, okay, there's a thymine here and it accidentally put a guanine here. That's incorrect, but we know that this strand here has methyl groups, so that must be the original strand. And so we know that this is the new strand, and so those enzymes detect that this one's the methylated one, this is unmethylated, therefore I need to replace the nucleic acid in this strand right here. And so it cuts out that section and replaces it. That's really, really cool. And then it has to religate those as well. And there's just a whole series of enzymes that actually do this, but they're looking for that methylation. So the proofreading ability of DNA polymerase and the mismatch repair ability of this other set of enzymes, those are going to fix any of these problems that get made by DNA polymerase. But we know that there could be a chemical problem, there could be radiation, and so we need other ways that can fix mutations that show up after both strands have been methylated. And how does that work? Well, one of those ways is called base excision repair. It's actually really, really straightforward. It just detects that there's a problem, okay? So in this case, it actually put a uracil next to a guanine. That's not good. And so all it does is you have a set of enzymes that come in, they sense that something's wrong, and they just cut out that one nucleotide and replace it. That seems pretty easy, right? So it's called base excision repair because it's just fixing that one base. Now, let's say you have a couple nucleotides that are messed up. Well, we switch to something which has kind of a weird name. It doesn't seem that different, but it actually is, called nucleotide excision repair. I realize that's kind of confusing because bases and nucleotides seem kind of the same, but in this context, they're different. So base excision repair is just fixing one nucleotide. If I use nucleotide excision repair, I'm going to fix a bunch of them at the same time. And we usually use this for UV light damage because it's not just one nucleotide that's damaged, right? We have a thymine dimer. That means two thymines are stuck together. And so in this case, we have two thymines stuck together, and that's going to be a really bad thing when your DNA polymerase or your RNA polymerase comes chugging along. And so we have a different set of enzymes that recognize that, and they say, you know what? If there's one place of thymine damage, then that means there could be a bunch of different thymines that are damaged here. And so why take that risk? Let's just cut out a whole bunch of nucleotides, and we'll cut out a whole bunch of them and replace those. And so that is nucleotide excision repair. It is a similar process. It's really just base excision repair on a bigger scale, i.e. 25 to 30 nucleotides actually get replaced, as opposed to base excision repair, which is a couple nucleotides, if not one. And so you can see between these two different techniques, you can actually fix a lot of the mutations that can happen. But I talked about earlier, free radicals or X-rays or gamma radiation, really high energy radiation can really mess up those backbones, and they can cause breaks in your backbones. That's really, really bad. And so we have to use a special set of enzymes and a special set of techniques to fix those problems. We call those homologous recombination, or we can use non-homologous and joining. I know those names are really long. It's actually super straightforward. And so let's look at non-homologous and joining. So let's say, for example, your DNA strands get hit with this huge dose of radiation because you visited Chernobyl because you're out of your mind. So your DNA is suffering from these double-stranded breaks. That's really, really bad. Well, non-homologous end joining, all it really means, the end joining part is the important part. And so what it's doing is it's just going to reconnect to the strands. That's literally all that's happening. And so they get glued back together. It's easier to glue strands back together that get kind of cut at an angle. And so you have a couple nucleotides kind of overhanging each side. And so it's easier to match those up, and then you ligate them. Now, a weird thing that can happen only during a certain part of the cell cycle, so you have to have undergone the S phase, is called homologous recombination. And that's a really weird one. And so in homologous recombination, what you're doing, and that's this over here, is you're going to take your sister chromatid. So we talked about like the cell cycle, and you have one copy of a chromosome from mom, you have one copy of a chromosome from dad, and when you need to go through mitosis and make two daughter cells, they each need one from mom and one from dad. So you double them, right? You have two from mom and two from dad, and one of those each is going to go into your new daughter cells. Well, if you have that other one sitting around and you have DNA damage occur, you can say, hey, I just made a new sister chromatid. Okay, awesome. Well, I'm just going to use that sort of as a template to fix this other chromosome or this other strand of DNA that got damaged. And I know this is a lot, there's a lot going on. And so what I wanted to do was show you a really, really good TED Ed video that gives a really nice visual depiction of all of these different processes I've talked about.
The DNA in just one of your cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times, nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of damage. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high-frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA backbone. Double-strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double-strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two complete double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available. Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve, but most of the time, we want DNA to stay the same. Defects in DNA repair are associated with premature aging and many kinds of cancer. So if you're looking for a fountain of youth, it's already operating in your cells, billions and billions of times a day.
So that was an awesome TED Ed video that I hope really gives you a nice visualization of the processes that happen. I like that one a lot because it goes into enough detail to give you the gist of what's happening, but it doesn't overwhelm you with like all the different enzymes and all that crazy stuff. Just know if you take like a genetics course with me, we spend a lot more time talking about how these actual processes occur. So I wanted to leave you with something to remind you that just because we're talking about mutations and mutation repair, that doesn't mean that mutations are always a bad thing. Yes, you could have beneficial mutations, but if you're someone like who has cancer, you may want to use radiation, or your doctors would want to use radiation to actually cause mutational damage in a tumor. So for example, let's say you have a tumor and your doctor wants to use radiation to cause single-stranded breaks and double-stranded breaks and really kill those cells. Well, your cancer cells are your cells and they have all these same mutation repair pathways that we just talked about, especially homologous recombination and non-homologous end joining. And so what you can do if you're a cancer is fix those same problems. Why not? And so what we do is we actually use drugs that block those repair mechanisms. And so that's what's being shown right here. And so, for instance, if you had non-homologous end joining occur, there are a couple enzymes called like BRCA that your cancer could use to actually fix that problem. And so we can use a type of chemotherapy to actually prevent those enzymes from doing their job, which feels a little counterintuitive. And yes, it can be really, really toxic. This is why chemotherapies can be really, really dangerous for people, because you're trying to kill part of the body, but not the whole body. So this can be really, really toxic, but it's better than dying from cancer. And so it's that, catch-22, a double-edged sword. But this is one of the reasons that I like talking about mutations and mutation repair, because it's really important to understand what's going on so that in the future, if you're a doctor or a nurse or something and you're working with patients who have cancer, they might be on some of these drugs and you need to understand what they might be able to do.